I'm so tired, man.
You tired? Why?
Well, I'm always having a misconception about DNA replication during class. I don't know what's wrong about it though.
You too?
Yeah.
I'm also facing the same problem. What is your misconception?
For me, I just thought that all kind of cells are actually using the same DNA replication process model. What about you?
For me, I thought that DNA replication occurs at just one location along the DNA molecule in both cells.
Eh, what is this?
What is that?
Replicopedia.
Wow, let's join this.
Alright, let's go.
잠시 후 두 번째 게임이 시작됩니다. 참가자들은 진행 요원들의 안내에 따라 이동해 주시기 바랍니다. 카메라를 바라보세요. 스마일!
Welcome to the moment where life makes a perfect copy of itself. This is the places where life carefully begins again.
Hey, what? Where am I? Where is I? Am I alone in this area? Whoa. What? What? Where in the world am I right now? Is this an organelle? Ah, I don't care about that. Let's explore this place first. Whew. Eh? What is this? A card? This looks like something was off though, Barry.
Welcome to this dimension. Now, you need to find the information about the replication if you want to leave the place. First clue. Go to the Hey!
How can you be here? Well, I don't even know actually where am I right now, but I got this card. It asked me to find some info regarding DNA replication.
Wow, a mystery card like this? Alright, then I can help you with that. Follow me.
Does the enzymes that involved in eukaryote and prokaryotes are the same?
Don't worry about that, I will gladly help you for that matter.
Well, they are not the same. However, both the enzyme have the same function. For your information, enzyme have the ability to speed up reaction and build up or break down the item that they act on. So, prokaryote have helicase, primase, ligase, single-stranded binding protein, topoisomerase, DNA polymerase 1 and 3. For eukaryote, they are also helicase, replication protein A, topoisomerase, primase, DNA ligase, DNA polymerase alpha, epsilon, and delta.
A few moments later.
Finally, I arrived at the nucleus. Oh, is this the DNA? There's so many of them. Wow, finally, I arrived at cytoplasm. It's so beautiful. The shape of this DNA, it's double helix, right? Pretty and cool. I wonder, how is Mr. doing right now?
My phone? Arif.
Hello, Mr.
Hello, Arif.
How are you over there? Long time no see.
Yeah, it's been a while. I'm fine here. Arif, do you want to know something? At prokaryote here, I see that DNA is replicated by using semi-conservative mechanism, where each doctored DNA molecule contains one parental and one newly synthesized strand.
Oh, it's the same thing with me at eukaryotes. I can see that the register fragments are actually initiated by RNA primer, which are removed by a joining mechanism.
Wow, that's totally similar with what I see here too. I also can see that they also synthesize the lagging strand discontinuously as Okazaki fragment.
That's insane! The process of it are quite similar, huh? Between the cell. But then, what is the difference of it?
In prokaryotic cell, you typically have a single sequence specific origin of replication. For example, ODC in E. coli on a circular chromosome.
Only that, in eukaryote, they possess multiple origin of replication per linear chromosome, allowing simultaneous initiations at thousands of sites to efficiently duplicate large genome.
Oh yeah, in prokaryotic cell, the process involves protein and simple regulatory mechanism, which the core include DNA A as initiator, DNA B as helicase, and the single-man DNA polymerase as polymerase 3.
Still, in eukaryotic cell, the process is more complex. It's involving large multi-protein complexes like the ORC, MCM helicase, and multiple DNA polymerase, and also additional factors like the chromatin remodeling and also cell cycle regulation.
Okay then, how about this? In prokaryotes, the Okazaki fragments are bounded between 1000 to 2000 nucleotides and processed by a simple set of enzymes. I am sure I am more superior on that.
Okay, the Okazaki fragment in eukaryotic cell are shorter in about 100 to 200 nucleotides, and their maturation also involves more enzyme and pathway, reflecting adaptation to chromatin structure. Alright then, let me excuse myself. I want to explore this place more. Till then,
Yeah, okay. Goodbye.
A few inches later.
So tired exploring this place. Huh?
Hello guys,
Welcome to the
Replication. DNA replication start with enzyme helicase. Enzyme helicase unwind parental double helix at replication forks. By breaking hydrogen bonds, the two parental strands can be separated and making them available as template strands.
Then, single-strand binding protein bind to the unpaired DNA strand, prevent them from repairing. However, when DNA is unwound, it becomes more twisted or supercoiling. Topoisomers help relieve strand of the replication fork by breaking, swiveling, and rejoining DNA strand by cutting certain place on one of the DNA strand.
After that, primates will synthesize an RNA primer at 5' end of leading strand and at 5' end of each Okazaki fragment of lagging strand by adding RNA nucleotide that are complementary, which mean adenine pair with uracil, cytosine pair with guanine to the DNA template from 5' to 3'.
Oh, what happened next?
Next, come to the DNA polymerase 3. This enzyme catalyzes the synthesis of new DNA strand by adding DNA nucleotides to the 3' end of RNA primer. Remember, nucleotides added are complementary to the DNA template.
But because of their antiparallel structure, DNA polymerase 3 can add DNA nucleotide only to the free 3' end of RNA primer or growing DNA strand, never to the 5' end. Thus, a new DNA strand can elongate only in the 5' to 3' direction. Upon replication, two strands will be produced, which are leading and lagging strands.
What? Leading and lagging strands?
Yes, in leading strand, DNA polymerase 3 will synthesize a complementary strand continuously by elongating the new DNA in the 5' to 3' direction toward replication fork. Therefore, only one RNA primer is required for DNA polymerase 3 to synthesize the entire leading strand.
Meanwhile, for lagging strand, DNA polymerase 3 will synthesize the complementary strand discontinuously away from the replication fork as a series of segments called Okazaki fragments. That's why more than one RNA primer is needed.
Wow, that's so cool. But how about the RNA primer earlier? Does it need to be removed?
You are absolutely correct. That is the job of DNA polymerase 1. The enzyme will remove the RNA nucleotides of the primer from the 5' end and replace it with DNA nucleotides which are added to the 3' end of adjacent fragment.
And last but not least, DNA ligase will join the Okazaki fragments together into a continuous DNA strand by forming a phosphodiester bond. Finally, 2 identical copies of DNA are produced. Do you understand the whole process now?
Yes, thank you very much. With all this information, I can finally get out of this place. See you later. Bye-bye.
I'm so proud of our work. Finally, we successfully preventing skill possessions among students. There's a lot of things that happen during DNA replication, and we learn a lot of new things from that.
I think what can I say about the process is that the DNA replication process is important in our biological process that happens in our body. It's responsible for accurate transmission of genetic information from one generation to the next one.
Exactly. Even if both prokaryote and eukaryote have their own contrasts and similarities on how the process works on, prokaryote and eukaryote have evolved replication strategies that reflect their structural and functional differences, which ensure the faithful DNA replications tailor to the need of each type of organism. Okay then, bye!
But wait, we have our next target. Let's go.